awesome job. So I don't want to spend any more time talking up here, but would invite up, I think we'll probably go in this order, right? Invite up Dan first, talk to us a little bit about impeded speed, and then Alyssa, Gabby, and Liam.
Yeah, dude.
And Dan, I can help you out here. We're
Frozen on the screen, so I will unfreeze you. And then you are here. And if we go to from current slide, good to go.
All right. All right. Imagine you're walking from the gym to tandem. Just nice and quiet, and then boom! A tiger pops out of the bushes and starts chasing you and wants to kidnap you and bring you to sup you. You sprint as hard as you can and just barely get away. But now imagine you're carrying your gym bag, or you're coming back from lax practice and carrying your lacrosse stick. How much slower would you sprint carrying a lacrosse stick? Impeded speed. How do lacrosse sticks affect sprinting in male athletes? We need to go back to where it all started. First, in the beginning, sprinting was solely a means of survival. Escaping crocodiles, bears, saber-toothed tigers, the quickest humans were most likely to survive. But then in around 776 BC, with the first Olympic Games, sprinting shifted to more of a means of sport instead of survival. Fast forward to today, and sprinting is pretty much only used in sport, and lacrosse is no exception. With the invention in the 12th century till today, sprinting has been a major role of lacrosse. So how does sprinting work exactly? Well, a few muscle groups work together in particular to maintain power and acceleration in the sprint and support and balance. And the power and acceleration, the ankle muscles, hip abductors, which is in particular the gluteus medius, the hip extensors, which are the hamstrings and the glutes, among other things, they all maintain power and acceleration, while support and balance is maintained by the ankle muscles, the calves, the knee extensors, which are basically your quads, and then the core. But in particular, the arms also play a massive role in sprinting, with them providing forward momentum and power in the acceleration phase. But after that, during top speed, the role of the arms is pretty unclear, and we don't really know exactly what they do. Could be balance, could be power, combination of both, something else, some X factor, we're not really sure. But one thing is for certain is that sprinting is powerful. In maximum all-out effort sprints, 2,000 to 2,500 watts of power can be produced in men, which for reference is enough to power 20 to 40 LED light bulbs for an hour. So, how was I inspired? Well, Coach McMahon's 4 pillars of sprinting. Shin angle, separation, which is knee drive, big strides, backward drive with the carpet, and hard arms. So, if arms play such a huge role in sprinting, what happens when you restrict that movement? My first idea was to basically tie your arms behind your back and have people sprint, but that's not practical, and I don't know about you, but the last time that happened to me was never. And then my second idea was to do sports equipment and see how that affects sprinting. So that transitioned to the lacrosse stick because it's easy to test, practical, and applicable to Williston athletics. So we're going to go to experimenting now. By far, the hardest part of my project was actually getting people to do it and recruiting people, which was really painful. I have included a text string below where I'm asking someone to do it at a certain time. They say no and want to do it at a different time. Then I say, can you please do it at the first time? And they say, no, we'll do it at that time. And then we ended up I had never tested them. So But another huge part of my experiment too was random assignment of sprint order for each participant. And what I mean by this is instead of having every participant run in the same order, say run with a stick, run without a stick, run with a stick, run without a stick, or run without a stick, run without a stick, run with a stick, run with a stick, that would eliminate, by making it random, that eliminates the confounding variable of fatigue. So if you ran both your non-sticks first and then your stick second, you'd be more tired for your last two sprints and thus run slower. So randomly assigning the sprint order for every participant on the data as a whole eliminates fatigue's impact. So the effect of the stick is more focused. I used my freshman year lacrosse stick, LePan's Dasher Blue lasers in the gym hallway for testing. Athletes or participants ran 20 to 25 yards in total sprint, with 10 to 5 yards being dedicated for acceleration. And then at the end, there's a 10-yard flying split. And the flying split allows me to measure top speed as well as time, which was very helpful for this project. And athletes would start in a 2-point stance, which I've demonstrated here. And then when you sprinted with a stick, you started with both hands on the stick. But once you released, you could really do whatever with the stick you wanted, but you just couldn't like drop it or like throw it around or something. And then also you might be wondering why it's 20 to 25 and not just a solid number. That's because 3/4 of the way through, I was using the tape that Coach LePan had marked out, but 3/4 of the way through testing, he moved the tape. But this only created more precise results though, because athletes had more time to get up to top speed. And then on the top right is a screenshot of what it looked like when you would get timing. It says dash. It would say flying instead. They're a little different. So these are some pictures from the actual testing. And the funniest picture award goes to Hennick. Now time for results. So basically, to get this histogram, I averaged every participant's non-stick sprints and stick sprints, and then I took the difference between those two results and then put them in this distribution. And what I found is that the lacrosse stick mean, or the lax player mean, was 0.016 seconds, and the non-lax mean was 0.03 seconds. And when taking the median, which eliminates outliers or potential outliers such as this value, the lax median was about the same at 0.01 seconds and 0.03 seconds. And this might not mean a lot to you, but what these numbers tell us is that 1, athletes ran slower with the stick, and 2, lax athletes were able to run less slow with the stick. And we can see this very clearly with the height difference in these two graphs. So there was a 1.38% increase in time for lax athletes, and then 2.87% increase for non-lax athletes. And then when we look at speed, the overall mean, or the lax mean, was less negative than the non-lax mean, and the lax median was also less negative than the non-lax median. I did the same, averaged the two speeds, the top speeds for athletes without the stick, and then with the stick, took that difference, and this is what came of it. And 4 people actually ran faster, but 22 people ran slower. And again, when we average out all the times, we can see that there's a 1.4% decrease in lax players' top speed and a 2.7% decrease in non-lax athletes' speed. Again, showing that lax athletes are able to run less slow than non-lax athletes. And a lot of what it said probably went in one ear and out the other for some of you. But what you need to know is that these results tell us that free arm movement and rapid arm movement is crucial to peak performance in sprints. Added weight and bulkiness noticeably decreases speed and time, or increases time and decreases speed. And then lax players are much better at running with a lacrosse stick, which isn't very surprising considering they run all the time with lacrosse sticks, so they likely have much better arm mobility with a lacrosse stick. And one thing I also got a lot is that a lot of people told me that it is very uncomfortable and awkward running with a stick, which makes a lot of sense. So one more very cool thing that I noticed that I had never thought about too when testing is that different athletes run differently. So in terms of arm movement, stride length, and lean, for example, all squash players leaned like very heavily when they were running. So what I'm going to need to do is I'm going to show you guys 3 clips of elite athletes, 1 gymnast, 1 cross-country race, and 1 sprinter. And then I'm going to show you guys 3 clips of participants that do those same sports. And I mean, if you know who they are, it's pretty easy to say what sport they do, but I'm going to have you guys raise your hands to say what you think they do. Okay, that's almost. So this is a cross-country race. You can see they run very tall, big strides. This is a gymnast, Asher Hong, and you can see he runs with his arms very low. I'm going to play that again. He's kind of like scooping. And then Usain Bolt. Kind of all know what he does. Okay. So this is the first clip. The third clip, I don't know what. Okay, well, they're not playing. Okay. So, raise your hand if you think this person sprints. If you think this person is a gymnast. If you think this person runs cross country. If you think this person sprints. If you think this person is a gymnast. If you think this person runs cross country. If you think they're a gymnast. If you think they sprint. Okay, well, you guys got it all right. It's pretty easy because you know who they are, but one thing, like, my favorite thing was seeing the middle person, Cole, and it was so noticeable when he was running, too, that he was running so low because he's a gymnast, and he was saying that's because when you're approaching a mat to do the spins and flips and whatever they do, you really need that force to elevate yourself. So why is all this information useful? Well, good arm mechanics will make you a better sprinter. If you train arms, or train arms specific for sprinting, you'll probably sprint faster. Arm specific training for sprinting, such as like loaded arm sprint training, putting weights on your arms, will make you a better athlete. Focused arm sprint training for stick athletes would be beneficial since you're carrying around a stick all the time, an added weight, added bulkiness. and that will be helpful. And also if Coach LePan, if you're a runner and Coach LePan is like making you do like arm exercises, trust the process because arms are helpful. And if you're being chased by a tiger, I would drop the stick, because even if you don't survive, you'll at least get to say you ran faster. And finally, Mr. Palicia, Ms. Russ, Coach LePan, my 25 participants, thank you for your help. I appreciate that about you guys, so thank you. Oh wait, does anyone have any questions?
We have time for one question if there were any in the audience.
Yes.
I'll ask you a question. So to me, this is, because this is like my space, right? I'm always using science to try to make our kids a little bit better athletes. But there's something that is glaring to me from your research.
Yeah.
That, and I'm trying to figure out how to word this question. That you can't improve from what we do in our space.
Yeah.
So, is there something to you that another piece that you're kind of like light bulbs off that's like, hey, if I want to make a person in any sport better, is there something that you're thinking? I'm trying to get you to answer what I already know in my head.
Yeah.
And it's not coming out clear, but there was something that was glaring to me that we can't cure from just making them more powerful, putting more force into it.
Yeah.
What would that be? Based off of your research, based off of everything that you've learned about?
I would just say like better mobility, better arm movement. So instead of kind of being more powerful, say like just more rapid movement, and kind of just more output, I get, like just efficiency. Like I also kind of don't know how to put this, but if your arms are also moving faster, instead of being stronger, that'll also generate more of whatever the arms do.
Sure.
That's what I would answer to that question. I'm sure you still know a lot more than I do.
To me, what it highlights the importance of sport practice,
Yeah.
right? Because the lacrosse players were better, even though they might not have more power output.
Yeah.
Right? They might not be stronger, right? But they're better at sprinting with their implement because they practice their sport.
Yeah.
Right? So you can make somebody more powerful and stronger, but you also have to be good at the skill.
Yeah,
Right?
yeah.
The skill of running with a lacrosse stick. Right?
Mm-hmm.
That's what I was thinking about. Sorry, I was trying to lead you to that, but
Yeah.
not a great classroom teacher.
Yeah.
Good. All right. Excellent. Thank you all. Let's rotate to Alyssa. That's right, Dan. Just make sure it's all right.
Thanks for coming.
Thanks.
Hi, my name's Alyssa. So as a kid, you probably told one of your friends or your bullies that sticks and stones can break my bones, but words can never hurt me. But what if I told you that words, sounds, can cause actual pain? So today I'll be tackling the question of does gender affect our response to sound and relating this to an auditory disorder called misophonia. So first, how do we hear sound? Sound waves enter our ear and travel through a narrow passageway called the ear canal, which leads to the eardrum. The eardrum vibrates from the incoming sound waves and sends these vibrations to 3 tiny bones in the middle ear. The bones in the middle ear amplify the sound vibrations and send them to a snail-shaped structure filled with fluid called the cochlea, which is the purple thing. The vibrations cause the fluid to ripple, which creates a traveling wave that forms along the inner membrane where there are hair cells, and the hair cells essentially ride the wave. So as the hair cells move up and down, the movement of nerve endings create electrical impulses that are carried to the brain by the auditory nerve. So this is how we turn sound into something we recognize and understand. So now that the sound is in your brain, how do gray matter and myelination play a role in our interpretation of the sound? Gray matter is responsible for sensory reception like seeing and hearing, and gray matter density actually increases from childhood to young adulthood. Gray matter, which you can see in the upper left photo, is actually gray because there's a high concentration of neuronal cell bodies, to give it its color. And one specific difference between females and males to do with gray matter is that females have smaller brains, but they actually have an increased density of gray matter, which is one possibility for why females have a comparable cognitive performance to males despite having lower brain volume. Now myelination, which you can see in the lower photo, the gray one is a non-myelinated axon, which is a portion of a nerve cell, and the pink one is a myelinated axon, is the process by which axons are covered by myelin sheaths, which allow for improved connectivity and neurotransmission. So myelin sheaths are insulating layers that allow electrical impulses to transmit quickly and efficiently along the nerve cells. So essentially, more greater myelination in one brain allows for higher and quicker processing. So to do with my research, here's the auditory disorder, which is misophonia. It's described as a disorder of decreased tolerance to specific sounds that elicit strong emotional and physical responses. For example, some people who have misophonia refuse to go out to eat with their friends because all they can focus on is the sound of them chewing, and that makes them really angry and disgusted, and they feel like they need to flee the situation. So this can be a debilitating disorder for some, as some misophonic patients have physical reactions to sounds that can cause them physical pain. So there's very little research on this order, as it's only been done for about 20 years. So some experiments include a more inclusive criteria for misophonia, while some include a less inclusive one. One more inclusive experiment said that up to 20% of the population has misophonia, with a range of how debilitating it is, which is a really large portion. And this disorder is not even classified as a psychiatric illness or a neurological disease, despite there being a difference in the brains of those with misophonia and those without. So misophonic patients have an abnormality in the emotional control mechanism of their brain and an increased connectivity between the frontal lobe, which is the front there, and the anterior insular cortex, which is highlighted. And also, something to note is that misophonia primarily develops in the early adolescence of females and affects more females, with one study saying that up to 83% of misophonia cases are female, and there's no clear reason why. And so I actually have this auditory disorder, which made me interested to figure out the why behind this and understand more. So one experiment done on misophonia was in 2017 at Newcastle University, which concluded that misophonia is an abnormal salience or recognition of particular sounds based on an abnormal activation and functional connectivity of the AIC, which was that portion of the brain I just mentioned. So in their experiment, fMRI data was acquired in misophonic and control subjects as they listened to multiple misophonic sounds like chewing and breathing, unpleasant sounds like a baby crying, and neutral sounds like rain falling. So the data showed that misophonic subjects had greater activation of their AIC when listening to trigger sounds like chewing, breathing, and snoring. So you can see some of this data in those three brain scans where the highlighted part is the increased connectivity in the AIC. So they also found that in the AIC where there's increased functional connectivity, misophonic individuals have altered magnetization transfer saturation, which basically just means significantly higher myelination in their gray matter of their ventromedial prefrontal cortex, another part of the brain. So this study actually provides basis for a structural difference between the brains of misophonic subjects compared to control subjects. So my experiment had many similar aspects to the Newcastle experiment, just on a high school level and with a twist. Rather than comparing misophonic subjects and control subjects, I compared females and males in the Williston community. So I had 40 participants total that were chosen randomly, basically me going up to random people in the library asking them to help me out. And there were 5 girls and 5 boys in each grade, not including students that repeated, so age did correlate to grade in this experiment. They listened to 11 different sounds and each played for about 15 seconds. So there were 4 unpleasant sounds, which were a baby crying, fire truck, nails on a chalkboard, and a person screaming. And then there were 3 main misophonic sounds of chewing, breathing, snoring. And then there was also water dripping and repetitive pencil tapping, which some studies said were misophonic and some said were just annoying. So those were kind of in an in-between category that didn't give me great data. And then there were also 2 neutral sounds of rain falling and the sounds of a busy cafe. So after each sound, the subjects were asked 2 questions. So now if everyone can take a second to listen to this sound. Okay, I want you to think in your head right now, how annoying was that sound on a scale of 1 to 10? 1 being not annoying at all and 10 being absolutely unbearable. Now, if you were in an environment and this sound was playing, how long would you be able to spend with this sound playing? 1 being forever, it doesn't bother you, and 10 being like, oh my god, get you out of there. So congrats, you were just a trial in my experiment. Each subject was asked both those questions, and their answers were recorded and averaged among the same gender group and the same age group. As you can see in these 2 photos of Ava and Bennett, the first 15 participants had their heart rate monitored on a pulse oximeter. However, I quickly found that this wasn't very accurate, as some heart rates were 200 and some were 15, so I unfortunately had to abandon this idea pretty early on. So interesting part, which is my results. First, for the unpleasant sound, the comparison between the total girls versus the total boys for the average results of their annoyance levels, which was the first question, and their fight or flight instinct, which was the second question, was negligible, which I considered about plus or minus 6%. So overall, they were the same unpleasantness to both groups. However, the comparison between the freshman girls to the senior girls, which you can see on the upper bar graph, the freshman girls had greater annoyance levels for the baby and the nails on a chalkboard than the senior girls, and for the screaming and fire truck, it was pretty negligible. And this suggests that they could be processing these sounds in a different way, potentially because they're younger. Also, on the lower bar graph, you can see that the freshman girls rated that they would flee much faster than the senior boys for all of the unpleasant sounds except screaming. So the positive is when the girls were higher, and the negative is when the boys were higher, just based on the equation that I used. And this suggests that the two genders process sound differently as well. Something else that's interesting during this experiment is when this sound played. Many of the girls actually told me they sympathized with the mom of the crying baby and felt bad, whereas majority of the guys rolled their eyes and asked to take off their headphones, which I found really funny. So also, for the neutral sounds, on the upper bar graph, you can see what a negligible difference looks like between the girls and the boys as they rated their annoyance levels similarly. There's only a 1% difference, so this shows just how small it was. And for the freshmen and senior girls were also very similar, with the younger girls only slightly more annoyed with the sounds, but not by a large margin. But something to note is that in the lower bar graph, you can see the difference in fight or flight instinct between the girls and the boys, and you can see that the girls were much higher, even though these sounds were meant to be like pleasant, calming, not bothersome whatsoever, so that was something that stood out to me. Now the data that I was most interested in was the misophonic sounds. So since misophonia primarily affects adolescent girls, I was curious to see if younger girls, or girls in general, had greater reactions to these sounds. So my data supports this claim because as you can see in the first bar graph, the girls rated the main misophonic sounds of breathing, chewing, and snoring significantly higher than the guys, but also in their fight or flight response, which I didn't show in the graph. And something that was interesting, which was kind of an outlier, is that between the freshman girls and the senior girls, the freshmen rated their annoyance of the breathing 26% higher than the senior girls, even though the chewing and snoring were negligible difference. So that was kind of an outlier that I thought was really interesting. And then on the bottom graph, you can see that when comparing gender, the freshman girls rated their fight or flight instinct significantly higher than the senior boys, and this was the greatest margin of difference I had throughout all of my data, so you can see how high that is. And so also there wasn't a clear correlation between age and response to misophonic sound when I averaged the data. And this could, as you can see on the top bar graph, that's for girls, and on the bottom, it's for guys. And there's not really a clear increase or decrease, it's just pretty flat. So this could suggest that misophonic responses are established before the age of 14 for both genders, because my age group was only 14 to 18. And if I had included a younger age range, like the middle school, I would have hopefully seen an increase in reaction to misophonic sounds once a certain age is hit. So my main conclusion is that girls overall rated the sounds more annoying and that they were more likely to flee than the boys, especially for the misophonic sounds. So misophonia is an auditory disorder, and the brain plays a role in processing sound. There's likely to be a structural difference between boys and girls that cause misophonia. So one possibility is their gray matter density differences. So since greater gray matter myelination means higher processing, it would make sense that people with more of it experience greater reactions to specific sounds, giving one possibility for why girls develop misophonia more than boys. And my experiment only shows that in the Williston community, although my participants were random so that generalizations can be made, that younger girls experience greater reactivity to misophonic sounds. So there simply needs to be more experiments done to help those who suffer from misophonia. So for example, people can monitor myelination in brains of those with misophonia and those without to draw connections between gray matter and age and gender. One way to compare gender is to see if all women have the same gray matter myelination and then see if boys have the same. If not, this could be a potential cause of misophonia. It would also be interesting to see if boys with misophonia have greater gray matter myelination than boys without to further support this claim. So maybe a treatment could be proposed that targets gray matter to reduce the symptoms of those with misophonia. And the question of why is yet to be answered. Why does misophonia happen? Why does it primarily develop in girls? Why do sounds, invisible vibrations, cause pain to some and not to others? Thank you.
Probably time for one or two quick questions. Anyone has any questions?
You talked about a fight or flight response?
Yeah.
In your study design, how did you define that, and then how did you measure it?
It was more on a scale of 1 to 10. So I told them that 1 was they would be able to spend forever in an environment with that sound playing, and 10 was that they would want to leave immediately. So I considered 10 to be a greater fight or flight response, and 1 to be like you didn't want to flee at all, so you had no response. So it kind of depended on the sound.
I saw that in your image, you had a little blood pressure or heart monitor on the finger as well. Is that something?
Yeah, so that worked for, like, I feel like for the first couple participants it did work. For example, when the screaming sound played, a lot of people's heart rate increased, even if the original one wasn't very accurate. But then as I kept using it, it's like it just kept getting worse and just didn't work once I even put it on some people's fingers. But in the original, in the first couple ones, it showed the heart rate increasing for sounds like screaming, and for some people, the chewing one.
Thank you.
I'm curious if any of the research you came across shows a correlation between head injuries and any increase in this type of misophonia in individuals, or if that's something that you think is worth pursuing as a study.
Yeah, that's not really something I focused on because I was more focusing on gender, but that would be something interesting because if you have head injury, if your myelination increases, and then that could also cause this person to have misophonia. So that would be something interesting to further research.
Excellent.
Hi, my name is Gabby, and today I'm going to talk about why I think that eating garbage might be the future. Basically, I think that using microorganisms as a way to decompose plastic pollution is the best strategy for improving our environment. So, okay, the big polluter is obviously plastic. So what is it? Plastic's basically a polymer made from crude oil. And some of the most common plastics are polystyrene, polyethylene, and polyester. They're really, really persistent and stable, which makes them really hazardous, pollutants. Probably the most staggering stat is that since the 1950s, when plastic was created, 8.3 billion tons of plastic waste have been generated, and of that, only 9% has been recycled. So plastic breaks down into microplastics, which are plastic pellets under 5 millimeters in diameter. Microplastics can be split into 2 groups, primary and secondary. Primary microplastics were created with the intention of being microplastics, whereas secondary microplastics have entered the environment and are broken down through physical weathering or chemical weathering. So their environmental impact. Microplastics come from things like cosmetics, packaging, agricultural products like pesticides, and they tend to find their way into our environment through water leaching through the ground into plants, organisms, water sources, all that. They have a super high surface area, so they can absorb a lot of toxic chemicals really easily. And so that makes ingestion really dangerous because when they get inside of an organism, they cause things like hormonal disruption, mutations, and even cancer. In terrestrial ecosystems, they can permeate plants, seeds, roots, everything. And that's really dangerous because when the plants get eaten, the microplastics work into the organism, including humans. So there are 2 big strategies for reducing microplastic pollution: advanced oxidation process and biodegradation. Oxidation strategies basically use light and energy to decompose the microplastics, but I focused on biodegradation, which led me to my essential question: how can microorganisms be used as a bioremediation strategy for microplastic pollution? So, what is biodegradation? It's basically when microorganisms use microplastics as a source of energy, resulting in decreasing microplastic weight, forming cracks, and making chemical bonding structures, and it releases organic compounds like CO, N2, and H2O. This is really, really efficient and doesn't produce any secondary pollutants, which is awesome, but it takes a long time. So how does this work? Basically, a biofilm forms when bacteria collects on the surface of a microplastic, which decomposes it into organic molecules. A subsection of biofilms is biofouling, which is when biomass settles on floating plastic, like algae in a marine environment. In a marine environment, this is called marine biofouling, and it also decomposes microplastics. So I researched some key experiments and bacteria. The 3 bacteria strains I focused on were, this is the only time I'm going to try to pronounce them, I'm sorry. Bacillus, Pseudomonas alcaligenes, and Alcaligenus fatalis. But all of them proved really productive on different plastic types and in different environments. So specifically, I studied 2 experiments. The first studied BC and AF and their effectiveness of 3 different plastic types: linear low-density polyethylene, high-density polyethylene, and polyester. And the second studied BC and PA and their efficiency at microplastic polystyrene degradation at their optimal environmental conditions. So in the first study, basically BC and AF were taken from soil known to be polluted with microplastics. They were introduced to a sterilized set of microplastics, and 3 strategies were used to measure their effectiveness. This was weight loss, the Sturm test, and surface modification screening. So the weight loss strategy basically introduced the bacteria into a soil coated in microplastic dust, and then measured the weight of the soil before and after the incubation period. And if it lost weight, it meant that the microplastic was being degraded. AF had significant weight loss for the 15-day period, but BC had like a huge productivity. You can see 30% decrease in 15 days is massive. And overall had a greater weight loss than AF. So the Sturm test, this is a picture of it. Basically, the bacteria was enclosed in a tube with microplastics, with all the plastic types, the 3 types. And the bacteria as the bacteria decomposed the microplastic, it would release CO2. The CO2 was then turned into a precipitate, and the weight of that precipitate was measured. Both BC and AF were able to produce significant precipitate, with BC producing 0.427 and AF producing 0.602 grams. So surface modification screening is probably the coolest. This basically took super up-close pictures of the microplastic after it had been colonized and affected by the BC and AF. You can see the visible cracks there compared to the control. All those dark spots were not there to begin with. So that's literally the bacteria breaking down the plastic, which is super cool to be able to see it. But I also want you guys, this is low linear density polyethylene. I want you to take note of how the BC and AF look different. They did not decompose the microplastic the same, which speaks to the specificity of the bacteria. This is high density polyethylene. You can see the AF has clearly decomposed it, and this is polyester, with again, visible degradation. So what does this mean? So basically, this experiment implied that bacteria can be used and visibly can show us that it's like that microorganisms can be used as a bioremediation strategy. It also showed us that there are differences in how bacteria break down the plastic. AF and BC both showed proficiency in degradation, but in different ways. For example, BC performed really well on weight loss, but AF was stronger for visible alterations in the Sturm test. This demonstrates that bacterial breakdown is by no means universal, and bacteria specialize in the plastic that they can break down. And this basically shows us that we need to expand on bacterial research, how things are breaking down, and how to study them so that we can get a more efficient bioremediation strategy. So the second study, this study measured BC and PA's ability to degrade polystyrene and what environment works best for the bacteria. It looked at pH, temperature, microplastic concentration, agitation speed, microplastic size, and the addition of glucose, which is thought to make the bacteria more productive. So the microorganisms were acquired through sludge, and the microplastics were acquired through grinding plastic spoons. Important factors were identified through trial and error, literally just testing the bacteria at a bunch of different temperatures, pHs, all that to find what worked best. And then the bacteria were put in that environment on plastic and studied for their effectivity. The results, generally CFU, or the ability for bacteria to reproduce, increased, and so did the carbon present. This strategy was super efficient and Overall, PA had a higher TOC and TIC value, so it has higher CFU and likely more function. That basically means it was really productive, and the high TOC and TIC values demonstrate that the bacteria was working well at degrading the plastic. BC individually decreased one sample by up to 58.8%, which is massive. And in 40 days, it generated a 7.4% weight loss, and its most important factors were determined to be low agitation speed and low microplastic concentrations. And then AF was able to colonize the polystyrene film. It showed a little bit less degradation, but its most important factors were determined to be high agitation speed and small microplastic size. So what do these two studies show us? Basically, they're showing us that decomposition is absolutely not universal, and specialization between bacteria and microplastic is very common. Bacteria can't decompose everything, and some microplastics are resistant to some microorganisms. There are studies that demonstrate that having a mixture of a bunch of different microorganisms together working on a microplastic is more efficient than having one bacteria alone working on a microplastic. And environmental conditions also play a huge role on how the bacteria are working, and environmental standards should be set for bacteria types. But ultimately, there are common characteristics, and there are constant ways to measure degradation that need to be emphasized and used. So environmental consideration. It's been determined that toxicity of microplastics does depend on environmental conditions. I focus on marine ecosystem for this section, but the effect of the ocean on microplastics is really different from in a terrestrial ecosystem because of their exposure to UV or lack thereof, which causes higher or lower toxicity. Or physical weathering in the ocean, which can create nanoplastics, which are even harder to break down. Microplastics are really affected by their environment, but obviously so are bacteria. So in one study, it was suggested that, opposite of the other two experiments I talked about, cold conditions might be best for degradation because it allows for the release of certain enzymes that are thought to be the actual thing breaking down the microplastic. So the environment definitely needs to be considered, and because it introduces a lot of variables that just can't be ignored. So my ultimate goal was to come up with a solution for microplastic remediation. And I came up with a 3-tier plan. Number 1, collection. So obviously water brings microplastics through our environment. I think we need to target landfills and water that runs through landfills pulling the microplastics out. That's called leachate. We need to collect that, and then wastewater, also pretty like laundry, also has a lot of microplastics. So collecting a bunch of water and then letting it settle, and microplastics will settle on the top or the bottom. You scrape off the microplastic and collect all this sludge from wastewater treatment plants and from your landfills, and you transport them to a facility with a chamber system. Place the sludge in a chamber, and this is going to be introduced to some bacteria types that all work at similar environmental conditions. They're subjected to these environmental conditions, moved on to the next chamber with different bacteria and different conditions that work better for them. Between each rotation, the CERN test is conducted to determine how much has been degraded, and before and after the whole process, the weight loss is used to determine how much has been degraded. And these are used as indicators, and then if it's degraded enough, it gets approved to move on to the third phase, which is compost, because there are fungi, worms, moths that have bacteria in their guts that can eat and break down the microplastic. So I think to fully get rid of the microplastic, the last step would be compost, and ultimately, hopefully it turns into a safe fertilizer to return the microplastics back to the environment. So microplastics are everywhere, but I think it's up to us to stop the harmful effects of plastic pollution, and I think that bacteria is the next step.
I'm curious if you read any studies about scientists editing the genes of bacteria that they know degrade microplastics and things like that, to see if they could maybe get it to where it works with more than one kind of plastic or even works quicker.
So I haven't read anything on that, but I think that's the next step, is genetic modification for bacteria. Maybe not for working on several different plastic types because they are all different, but I think the next step would be improving efficiency. Like an idea that I floated around for my solution was introduce them to a catalyst, which is basically putting the microorganism on steroids and making it work more efficiently, I think is the next step rather than having it work on several plastic types. Yeah.
Great job, Gabby. I'm curious to know if you have suggestions, since I don't have the ability to go filter sludge and wastewater and do all those things right now in my life. Are there things that people can do now to help sort of prevent some of these ideas, or rather prevent microplastics from becoming such a problem in our environment?
I think the best thing you could do would be proper disposal of plastic, because a lot of microplastics are secondary microplastics, and so that's a massive issue. As for actually individually taking microplastics out, I don't actually think that's possible right now, just because they're everywhere at this point. I'm breathing in microplastics as I speak, so they're so everywhere. I think trying to individually reduce microplastic use is like almost pointless. It's all about the grand scheme of things and overall better disposal of plastic products.
Did this project make you want to continue researching this topic in college?
Yeah, I want to go more in depth. I don't feel like I know enough about this. So.
Nice job.
Excellent. Nice work again. Scholars, I would ask that we stick around. If you're a few minutes late to advisory, I can send your advisors a quick note. Is it advisory check-in? Yeah. I think if you're a minute late to that, I'm going to try to give them our full attention.
Okay, well, I'll be quick then. So,
Please take the time. Please take the time.
this is you, and you love sharks. But maybe this is a little too close and perhaps on the nose, and maybe you don't really like the aquarium setting. So I think I propose a better solution. My name's Liam, and I'd like to introduce you to my Scholars project, Sculpting Sharks. There's an animation coming, I promise. So what is my project? This fall, I've been creating a 3D photogrammetric model of an epaulet shark. I've been researching the kinematics and locomotion of these animals, and I plan to animate the 3D model in the future. To give a little background, I'm an intern under Duncan J. Urshick at UMass Amherst in the biology department, and in our lab, we focus on creating 3D models of animals and researching them to provide free 3D models online via a website called Sketchfab. And these have a plethora of uses, from conservation to teaching to just recreational use, whether you're creating like an animation or 3D printing something, you can use our research. And so over the summer, we had the privilege of working with Mystic Aquarium and going down to Connecticut and photographing and videoing some of the sharks they have on site there. And my fall has been spent 3D modeling and researching a specific male epaulet shark that they have on site. And in the future, this winter, I'm going to animate the model and publish it. But before we get too deep into the details, I want to share the question behind my research and kind of what's driving this project. And by digitizing and animating a 3D model of an epaulet shark, what can we learn about the kinematics and locomotion of these animals? Because currently, kinematics and locomotion research is not really ongoing, and we don't really know anything about how these creatures move and why they move the way they do. But before we get all in the weeds with that, you all are probably wondering, what is an epaulet shark? They're called walking sharks. They literally walk on their pectoral and anal fins, and they are able to go into low oxygen environments and stay out for hours on end, out of the water, and this is to get prey, like small fish and little crustaceans. They live in shallow tropical waters around northern Australia, New Guinea, and also ranging into Indonesia. They're found in coral reefs, seagrass beds, and mangrove areas, and while they are primarily nocturnal, during the day they're often found in calm, shallow waters, hiding under rocks or coral. Some physical characteristics: they have a very distinctive dark eye spot, or an epaulette, on either side of the body, hence the name epaulette sharks. You know, biologists are very creative in their naming. They have a small cylindrical body with a short snout and two small pectoral fins. Those are the two right there in the front. And finally, they're small to medium in size, about ½ meter to 1 meter in length. So what does this mean, and how do we use it to make a 3D model of an epaulette shark? Well, we use a blender. Well, sorry, that's the wrong thing. But we use Blender, a open source free 3D modeling software. And we use a process called box modeling with image reference. And essentially, I'm going to walk you through some of the steps and try to give you insight into how we do this process. So you begin with reference images, dorsal, top-down view, and lateral from the side view of the shark. These are ones from the Visit to Mystic, from videos we took of the epaulette shark, and it's crucial to stitch together one image so that you have a straight shot of the shark. As you can see, stitch marks on the bottom image, because there was no clear nose-to-tip image that we could capture. And then next, you'll align them in Blender to create a box that'll reference for your modeling once you add a cube to the scene and center it with the axes. Now it's visible in between all the reference images, and you can begin to start moving and scaling the box until it encompasses the silhouette of the creature. As you can see, I switched sharks. I swear this is still my work. This is other work we've done in the lab with the same data set from Mystic. It was a clearer and easier process, and I figured that would give you a better insight into what we do with box modeling. And so you'll begin with, once scaled, you'll begin subdividing the model. You can see those lines. We're beginning to cut the cube as if it's like a loaf of bread with a tool called the loop cut. And each cross section is an edge loop. And by making a handful of vertical loops, we begin to start to see the shark take shape. And the more we make, the more we'll trace the silhouette of the reference image with each edge loop. Before we continue to make further details, we'll align an edge loop with the start and finish of each fin. And it's crucial to do this before going into further subdivision, because it'll make your life a lot easier. And after doing this process on the top and bottom, it starts to look like a shark or maybe a blob, depending on how creative you are. But then a little bit later, we're back to the epaulette, of course, it starts to kind of take shape. Now it's a blob with fins and an eye. And it's a lot of poking and prodding and pushing and pulling until it starts to become smoother and look more like a shark, more organic. And then eventually, after a lot of work, you're left with this. This is the final 3D model of an epaulette shark in all of its glory. And just to kind of recap, we went from a cube to this, an organic shark. And it's a time-consuming process. It takes a lot of patience, and just to kind of show you the level of biological accuracy that we intend in the research here, I have some side-by-sides. Here's the ventral view of the model. That's from the bottom. The dorsal, the face, and for reference, this face alone, the mouth took me a week to do. The head-on image, and yeah, so this is kind of like where we start, right? We compare, and then we do our best to like recreate the shark in Blender. So what is the future of this project? In the immediate future is coloring and texturing the model. The minute we release this to the public through Mystic, we'll begin to give it basically hair and makeup. And then the next step after that will be animating, so kind of combining the research I've been doing in kinematics and locomotion with actually animating the model and making it come to life and move. And then finally, publication. In the Urshik lab at UMass, we have a data set of around 20 3D models of sharks, and we plan to publish a paper with all of them demonstrating the importance of 3D modeling sharks and why we should continue to do it and how it's crucial to the scientific process. So, lastly, you must be wondering, why is this important? You played with digital clay, great. But let me tell you, so, you know, as the oceans begin to face more threats with climate change looming, it's crucial that we preserve creatures like this because we don't know how much longer they have a habitat, and they could be threatened at any moment, honestly. And they're so beautiful. It's really key. It's crucial that we keep them alive digitally. And then also we're able to learn more about body condition, locomotion and kinematics, other unique behavior and survival tactics through this digitization process, right? So for example, the walking and the being able to be in low oxygen environments, we can learn more about that as we learn more about how they move and why they move. And then lastly, much of science is kind of filling holes in what we already thought we knew, and fixing processes that are old and not correct, right? So maybe we think that epaulets walk because they like the sunshine, right? But we need to fix that hole in our knowledge to set up a more efficient future for epaulet and general shark research.